And we applied to the United States FDA and we were awarded a phase 1/2A, 9 outcome measures of efficacy and about a dozen outcome measures of safety. This is a first in human test, and it's been going on over the last year at the University of California Irvine Medical Center, UCI Health. And I'm just absolutely delighted to say that in older humans that are immobilized, so they can't exercise. This is immobilized elderly humans. We're seeing their muscles grow. We're seeing them get stronger. The muscles get stronger and the whole limb getting stronger. We're seeing in a functional quality of life measurement recognized by the FDA and CDC as a quality of life measurement. It's called the 6-minute walk test, that they are statistically significantly improved, 20%.
Hello, Dr. Keirstead. You are the founder and chairman of Immunis Biomedical. So a warm welcome to Modern Healthspan, and thank you so much for being with us today.
A real pleasure to be here. Thank you for having me.
So Dr. Keirstead, so I believe you're a neurologist by training, and then you went and studied stem cells for a while. But now most recently, you founded Immunis Biomedical, which is focused on the immune system as a driver of aging. So can you tell us a bit about Immunis and why you picked the immune system kind of as your latest target?
Well, my prior most recent company is Avita Biomedical, where we had developed a treatment for cancer, a personal immunotherapy for cancer, run it right through to phase 3 clinical trials. And I feel so fortunate to have been at the helm of a story that worked so well to save so many lives. So that's quite a mature story, just being, in the middle of deals, licensing it off and that type of thing to big pharma. And really excited about where it's going to go and where it, how it's looking. But it occurred to me that coming off of that story, that, a necessary precondition for 100% of cancer patients is a suppressed immune system. And it really, over the years, really got me thinking a lot about developing a modulator of the immune system to stop immunosenescence that occurs in 100% of humans. And at Immunis, we've managed to do that.
Right. So, yes. So when we look at modulators of aging, you said that the underlying mechanism is immunosenescence in many cases. Could you get into that in a bit more detail?
Sure, yeah. The immune system in many ways is the primary controller of your aging. It's just a fact that most of the manifestations of aging are actually immune mediated. Muscle atrophy, metabolism, arterial stiffness, neuroinflammation, wound healing, the loss of the adaptive immune response. These things are all immune-mediated manifestations of aging. And the importance of the immune system in your health span and your lifespan can't be overstated.
So how does the immune system change with age? What kind of changes do we see?
Basically your immune system gets old and angry with age. It's 100% of humans develop pro-inflammation, just a chronic state of inflammation within their blood systems. And that is largely mediated by the fact that you have fewer progenitor cells. The young cells of your immune system are born very frequently when you're young, and they're born very infrequently when you're old. And as it is the young cells, the progenitor cells that secrete, you have fewer secretions in your blood, because when you're over 27 years old and you're birthing your immune system less frequently, you have a dearth of these, what are referred to commonly as nursing factors, hundreds of growth factors and cytokines and chemokines, exosomes, a few other things that are released by young progenitor cells. Your mature cells do not secrete. It's your young cells that do. And because you birth your immune system less frequently with age, you have fewer progenitor cells of your immune system, and therefore, with age, you have fewer of a secretome, these nursing factors. And what those things do is DNA repair. They are responsible for rolling of the immune system out of your blood and into tissues. And that's a big deal. 80% of your immune system resides outside of your blood. So these extracellular matrix and cell-cell adhesion molecules and cytokines and chemokines and a few other things that cause the rolling and the attraction and movement of these immune system cells, you just have a lot less of them with age. So at Immunis, we developed a method of taking human stem cells and making them for the first time in the world to a young, defined progenitor cell population, human, of course, that makes 440 active factors that modulate the immune system. Just like your immune system precursors made tons of it when you were young, we can basically make that in huge bioreactors now by the billions. Basically take young human stem cells and then make them into these young progenitor cells and define their differentiation perfectly so that we have a secretome that is identical batch to batch. And that secretome is every factor relevant to human immune system development and health, the very natural factors that you're full of when you're 27 and you have quite decidedly less of those things as you age. We found a way to make them. And in both animal models and now, super exciting, in humans, we see that it's both safe and efficacious.
Right. And would love to dive into some more of that in more detail.
Happy to.
Yeah. So the immune system is kind of going down with age, it becomes actually, it becomes more, it's like you said, angry, right? So we
Yeah,
get
like
this.
I'd phrase it as functionally deficit, but hyperactive. So it
Right.
is actually pro-inflammatory. It's freaking out, but functionally, therefore, it doesn't work very well.
Right. And so the best marker. So the question is, how would you tell kind of where your immune system is? Would you be looking for markers of inflammation?
Yeah, definitely. And as people age, there are certainly biological clocks that have done a good job of tagging particular markers that correlate quite well with immunological function and aging. Basically, what they are looking at as you age, there you can tell that the immune system is getting older because there's generally a lot more pro-inflammatory markers. And then there's an increase in your memory T cells and a decrease in your naive T cells. Your naive immune system, think of it like, as the word says, youngster, eyes and ears open. But when that youngster encounters a bug like COVID-19, it switches from naive into a memory T cell after, of course, it may evoke an immune response against the bug, but you remember it. So your naive immune system is actually depleted with age, and then filled up with a whole bunch of memory of all of the bugs that you've experienced, that type of thing. So you measure the amount of naive cells and the amount of memory cells and the amount of pro-inflammatory markers. And with these three general categories, you can do a very good job of biologically aging the immune system.
Right. Okay. Yes. And then the lack of the immune system is one of the key drivers of cancer, although, I mean, it has lots of other negative impacts as well, I guess.
Correct. Yeah. Your immune system, you think about it, it touches every single organ in your body. What else does that? You've got nerves that reach out to your organs, but your immune system is responsible for keeping that clean.
Mm.
Every organ of your body to fight bugs, invading pathogens, even messed up cells that you make. You replace your body about 15 times in its entirety throughout your life. That's a lot of cell birth and death. And it's your immune system that's doing a lot of the work in tidying that up, cleaning that up, making sure that it doesn't break down and get full of too much garbage and recycle correctly and all that type of thing. So your immune system is very active, both in keeping your body working well, but also keeping it clean and free of invading pathogens.
Yes. Okay. So Immunis has is it a product or a technology called Immuna, correct? It's
It's a
a tech,
product. Yeah, it's
it's
actually, it's a new drug in a
right,
brand new drug category. It's called Immuna.
Immuna, right. Okay, so could we talk about so you said you're using human stem cells, young human stem cells. So can you talk about
Yep,
where are you getting them from and how are you
sure.
creating so much um, secretome from them?
Yeah. So we use a ethically procured, laboratory generated, NIH um, registered. These cells have provenance. The starting materials are either iPS inducible pluripotent stem cells that are human or embryonic stem cells that are human, obtained from the discard of fertility clinics. The the world is you know split in some jurisdictions really like ips cells, some jurisdictions really like embryonic stem cells, so we can make the product out of both of them.
Interesting. And so stem cells can just keep generating. They don't hit like the Hayflick limit or anything like that, or do they?
They do. Yeah, not all stem cells are equal. Embryonic stem cells divide the best. So they are the most commercially viable of any cell type. They are not procured from aborted fetal tissue in the back door of abortion clinics.
Mm.
This is not fetal tissue use. This is the discard of a fertility clinic where a woman superovulates and takes many eggs. The man donates sperm. Too many eggs are fertilized, and the one usually gets put back into the uterus for in vitro fertilization, going into a fertilization event for the couple. It usually works nowadays, leaving a few eggs that are left over, and after several years, the owners of them usually just discard them. If a stem cell researcher wishes them, they can consent the owners and say, "Could we use these?" And otherwise, they're destroyed. And the human embryonic stem cells are pulled from them and used for generating treatments and immunotherapies like this one.
And how much treatment can, I mean, how often do you need to renew like the stem cells?
You really don't. It's
Yeah.
they like a an embryonic stem cell divides very, very well. It's naturally occurring, it's very young, and once those things are isolated, they can last hundreds and hundreds of years of dividing. You could, in a single stem cell line, if you didn't stop it from growing, the earth would be covered in about a 12-foot slurry since they were first developed in the late 90s, or isolated, I mean. But you know,
Mm.
uh, uh, iPS cells, when you take human skin and then bombard them with a whole bunch of factors that
Mm.
genetics that are expressed by young cells, kind of tricks the skin cell into thinking that it's a younger embryonic cell, and it back differentiates and becomes a young stem cell. So it's another way of making it. But the
Mhm.
means of making it, jamming in 1,500 copies of four different things, beats up that cell. So the cell is a little less capable of dividing. And so it'll stop. It might do 10 or 20 divisions and then start to differentiate, and you lose your pool. Whereas embryonic stem cells have a much more robust ability to stay young, and therefore the banking of them is really one cell has really, you know, formed the foundation of almost every treatment that my laboratories have ever generated in the last 30 years.
Do you know magnesium is a critical mineral for cellular function? Perhaps the most important role is being bound to ATP, the currency of cellular energy, as magnesium ATP complex, which is the recognized form of the cofactor required for proper function by hundreds of enzymes. Research suggests that magnesium deficiency can contribute to increased oxidative stress, accelerated cellular aging, and mitochondrial dysfunction. To make sure that we are not deficient and to support optimal mitochondrial function, my wife and I take magnesium breakthrough daily. Unlike some supplements offering limited forms, magnesium breakthrough is made of all natural ingredients and contains a full spectrum of 7 forms of magnesium to ensure well-rounded support for your cellular health. If you want to boost your mitochondria, simply go to bioptimizers.com/modern and use promo code modern10 during checkout to save 10%. Thank you for your support. And actually, you're not using the cells. So you keep the cells. The cells are like a factory that are generating the secretome, and you're extracting the secretome. Would that be correct?
Yeah, that's exactly right. It's very interesting. We are, in a brand new drug, in a brand new drug category. It's very fun at Immunis. The 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 cells that we start with are stem cells, and then we make these young precursors that secrete this immunological modulator soup, if you will, of 440 factors, the secretome. And we just keep farming the cells, but the product is actually just the proteins, the secretome, what is secreted. So there's no self or non-self on those things. They don't get rejected. So therefore, this is off the shelf. It is something that you can just use from anybody because your immune system factors are like mine. My TGF beta is like yours, my IL-10, my IL-2. They're so similar across genders and races, ethnicities, that we can actually make them. It's called recombinant proteins synthetically and use them across genders and ethnicities. So the getting them from one particular cell line is good. Basically, they're not rejected by any other individuals. So it's off the shelf. It's a secretome that has 440 active ingredients that targets the immune system in 440 different ways. And that is really the special bit of what we're doing. The immune system is so complex. It is so incredibly complex. I'm a neuroscientist. I thought the brain was complex. Started getting into immunology about 15 or 20 years ago. And my goodness, it is so, so complex. For every physiological thing that your immune system does, there's 5 or 50 things pulling and 5 or 50 things pulling. It's like an orchestra that has a beautiful, harmonious output, but it's a lot of work to get that output. And drugging that with a single active ingredient, like every drug you've ever taken in your life, every drug is a single active ingredient. But back in the days when botanicals were coming up and lots of good medicines were being made from plants, the problem that the FDA had at that time is batch to batch variability. You boil a pound of willow bark and you get a different amount of aspirin derivative every time. So it's hard to make. It's sort of like a terroir in wine. So as you know, botanicals are not generally regulated by the FDA because of the batch to batch variability. Whereas cells, the developments in cell culture have been happening in leaps and bounds over the last 20 years. My laboratory at the forefront of this. And we're perhaps the world's best at growing homogeneous populations of stem cell derivates, meaning we take stem cells, make them into something in purity. Oligodendrocytes, a spinal cord cell type that's missing after spinal cord injury. You make that in purity, and it restored, quite a lot of function to quadriplegics in clinical trials. We've made high purity cardiomyocytes and neurons and neuronal progenitors. And in this case, a cell type that's young and secretes every factor relevant to immune system development and health. It's an interesting new drug category, the secretomes. Just a few years old. I believe that Immunis is only the second company in the world that's making them, or in the United States. Excuse me. And 440 actives in one drug.
Yes, that's really interesting. So there were a couple of papers that talked about using exosomes, so young exosomes, to rejuvenate other animals. And that was actually cross species, which was a bit strange. But how much of the content of the secretome is the exosome, and how much are the other functions, the other proteins?
An exosome content of a secretome is generally 0.5%.
Right.
Exosomes have been a kind of a new thing in the field. They're pretty amazing. They're little bundles of genetic material that have to get out of a cell and then go across a space and get into a cell, dump their contents, and then alter the genetic expression of the recipient cell. A lot of steps in there. Very
Hmm.
different from like a little protein that's released by a cell. You call it a growth factor. And that growth factor just simply has to, you know, diffuse a little ways. When it hits the outside of the new cell, it hits a receptor and boom, triggers an activity. So exosomes have more steps, but they do different things. So they're both very good. Immuna contains both.
Right. So do you take the complete do you take the complete exosome secretome?
Yeah, we
So
take the whole secretome. It's you know, it's the truth that we simply don't know what the active factor is.
mhm.
We have a really good idea from literature, like immune modulation of muscle is done by follistatin and fetuin, and playing a big role in that. And those are two of the highest expressing components in our 440 component secretome. Um, to knock them in, knock them out, investigate which 5 or 50 of the 440 are doing the real job is, in our view, secondary. I've had a thesis, a foundational philosophy, if you will, being a stem cell researcher in my whole career, and that is mess with the system as little as you can. Life is way better than us. And, when you try to simulate the brilliance and artistry and mastery of the human body, you fail. There's no scientist that can keep cells alive forever like the human body can. There's nothing like this vessel of ours. So as a general thesis, mess with it as little as you can. These young cells that modulate our immune system are already doing their job, and how they were programmed to do so, how their epigenetics is altered. They're doing a really good job of it on their own. Let's just capture them and let them do the job. So we are working with the right defined cell population that makes 440 factors that are identical to what you have in your system. So we've got a system here that's already doing the job for us. We just had to do the high purity derivation bit where we feed stem cells, push them along a developmental path. They want to become every single cell type in your body. It's very hard to control them, but we've gotten good at it over the years, and we are the leading group in the world doing this. And in this case, we made the right population for modulation of the immune system.
Right. That sounds like a great way of looking at it, I must admit, because it is just so complex. Like just
Yeah.
tweaking one little
Yeah.
thing at a time just doesn't work.
Yeah, like think about if you had to think of what 440 factors need to be secreted together to modulate your immune system. I can tell you for certain that we as a community, all immunologists together, the entirety of immunological knowledge on the planet does not know that. We don't
Mm.
know. It's so in this case, we're letting biology do it on its own. You know, I'm the chief executive officer of the Human Immunome Project. It's
Mm.
the world's largest immune endeavor. Multiple countries all working to decode the human immune system using multiple, very, very modern techniques of assessing the immune system. I don't know of a single immunologist that has used all of these techniques. They're all cutting edge, new, some of them exceedingly expensive. And we've gathered them together in what's called an immune monitoring kit and are deploying that this year. We're beginning our deployment into millions of people of all ethnicities, of all socioeconomic classes, all genders, of course, and decoding the immune system. But I can tell you for certain the reason that that needs to be done is because we know so little about it. So this thesis of Immunus was to let a naturally occurring biological system, let it run its course.
Yeah. So gave your you gave your product to some people to study, like preclinical, I believe. Could we talk a little bit about what you saw, and I
Sure.
guess yeah,
The
what you
fun
saw
stuff.
on, yes, the fun stuff, and
Yeah.
how they deployed it, I guess.
Yeah. Well, we began in mice
Hmm.
and now humans. And I'd be very happy to talk about both of these things because it's all quite new. I'm at a meeting right now. I just got off a stage at a big scientific conference in San Diego speaking about this very thing. So it's a lot of fun to talk about. With the rodents, what I did, being a professor of some renown, I've had a history of being a professor 15 years at the University of California, Irvine, and Keio University in Japan. And graduate students and assistant, associate, full professor, chartered member of the NIH, 150 peer-reviewed pubs, lots and lots of professorship under my belt in my history. And the uh that led me to gather a team around myself that is, pretty phenomenal, and published 150 papers, as I say. So I can now go out to professors of some renown in their sector that I know, or if they don't know me, my CV is good. They know that I've published good work, so they don't mind working with me. So what I did is I reached out with my team to 7 of the leading researchers in the world, each one of them on a different manifestation of immunological decline. So one professor, one of the leading scientists in the United States on muscle atrophy, another one on arterial stiffness, another one on adaptive immune response, wound healing, neuroinflammation, metabolism, etc. All of the major manifestations of aging, all of which we know are immune mediated. Because we have a multi-active immune modulator, I wanted to test it out on every animal model of human age-related and immune-related decline. And in all cases, we saw phenomenal benefit. We ended up grabbing onto muscle atrophy and metabolism for a couple of good reasons. One is that there's no competition. There is no approved way of changing muscle mass and gaining muscle mass. There's no drug that does that. Metabolism, we've just seen some pretty remarkable metabolic modulators recently with the GLP-1s and Ozempic, shrinking fat, but they're also shrinking muscle. That's very bad. So, we honed in on, huge market, but also driven first and foremost by dire medical need, and started working on rodents with muscle atrophy in a big way. So these are aged animals and also immobilized animals. Think about having a cast, you lose your muscle. So either immobilization or age are two things that for certain, without a doubt, no escaping it, you get muscle atrophy. So in these models, when given our drug Immuna, either the immobilized or the aged immobilized, they actually grew muscle and they got leaner and stronger. Their whole body coordination was better, right down to the cellular work of myotube fusion and increasing by 2 or 3 times the amount of young satellite cells. These are the cells that have to grow to build muscle. You take Immuna and you get more of it, even right down to the genetic expression patterns that change in order to do that. So right from, it's soup to nuts. It's right from the genes to the cells to the size of the muscle and strength of the muscle and balance and coordination. And in aged animals, Immuna certainly reversed metabolic and muscle atrophy. So I just ran over muscle. The metabolic was just also fun to see. Professor Drummond has these very expensive metabolic cages, and you put mice in them and they measure everything. Well, the amount of activity of these mice is about doubled. So for some reason, the animals are just moving a whole lot more day and night, double. They're consuming twice as much oxygen, excreting twice as much CO2. They're eating more, they're defecating and urinating more. When you look at their fat, it's reducing in size as well as cellular. The number of cells in the fat is decreasing as well as the whole fat. Pads are all decreasing. Fatty liver decreases. The toxic lipids of one's metabolism, these are biomarkers. As we age, our metabolism gets poorer, and we get a lot more toxic lipids in our bloods. These are things called ceramides or triglycerides or diglycerides. And as you're older, you have more of them. In the aged animals, they dropped down to about half when they received Immuna. All of this metabolic benefit in the very same animals, they're also gaining lean muscle and having all those muscle benefits, balance and coordination that I mentioned. So we were extremely enthused. These two works are published in very, very good journals. Aging Cell just accepted and is in print for the metabolic work. And Geroscience published just late last year our muscle work. So very good journals, impact factors of 8. About 95% of journals have an impact factor of 1 to 3. So these are very good, it's good work. And then we applied to the United States FDA and we were awarded a phase 1/2A, 9 outcome measures of efficacy and about a dozen outcome measures of safety. This is a first in human test, and it's been going on over the last year at the University of California Irvine Medical Center, UCI Health. And I'm just absolutely delighted to say that in older humans that are immobilized, so they can't exercise. This is immobilized elderly humans. We're seeing their muscles grow. We're seeing them get stronger. The muscles get stronger and the whole limb getting stronger. We're seeing in a functional quality of life measurement recognized by the FDA and CDC as a quality of life measurement. It's called the 6-minute walk test, that they are statistically significantly improved, 20% faster and further walking within 6 minutes. Their pain is dropped 50 to 70%. Their immune systems, the chronic state of inflammation of their immune systems is dropped by about half. That's extraordinary. 3 months after treatment, 3 months after treatment, these 60 and 70-year-old people have an immune system that is characteristic of someone decades younger than them. Their basal state of inflammation is dropped dramatically. So it seems to be working and no side effects, which is wonderful. So this is not published, the human work yet, because that just rolled out over the last month. So we're collecting all of the rest of the phase 1/2A data through to August of this year, when it'll completely end. And we have prepared a phase 2 already because the data are without any side effects, no adverse events, and we're having tremendous efficacy. We have already gone ahead and written the entirety of the phase 2 clinical trial, in which we're going to test both muscle and metabolism now in a much broader range of humans that have a broad spectrum of metabolic and muscle disorders. This is called a basket adaptive clinical trial that has a lot of people in the beginning, and it's expensive because there's a lot of people, but it's de-risked because there's a lot of people, and you can adapt it to the responders in order to ensure that you're really focusing on what works best and giving humans that, but you're also taking a broad look at many outcome measures in order to try to capture as many benefits as we can. And as I've relayed, the benefits of a healthy immune system are diverse. So that means we need to capture a very broad set of inclusion and exclusion criteria. It's pretty amazing. It's a lot of fun.
Yes. So what does the application look like? Is it an injection, and like how often? What kind of dose?
Yeah, right now we are just maximizing the opportunity for signal by injecting twice a week for 4 weeks into one leg, the quadriceps. And these individuals can't locomote, and it's because they have knee osteoarthritis, so they're have very limited locomotor abilities, and they're older, so they're naturally sarcopenic, which means muscle atrophy with age. So they have two things causing their muscles to shrink, and they lose a lot of muscle very quickly during that period of time. So we want to just inject one leg only in order to maximize the chances of seeing an effect. After that, of course, we would broaden it out to perhaps subcutaneous needle injections. But for a first in human, we're trying to maximize it by concentrating it into one large muscle group. So we pick the quadricep, and we see a beautiful signal there. But it's important to recognize that the muscle is like a leaky bag. It's highly vascularized. So whatever drug you put into a muscle, it goes everywhere pretty quickly. That's called systemic, your whole system. So, like in the mice, we're also seeing a systemic effect in the humans. And that's a good thing in this case because
Mm.
a modulated immune system is healthy for an old person. It's very healthy for a young person. We get dysregulated with age. This is helping it get better, demonstrably decreasing high levels of pro-inflammation in these humans. So we know it's leaking into the system, and that's good. That's not a bad thing, but it is concentrated. So we are seeing concentrated effects in the leg. Like, for example, the muscle growth in the injected leg is bigger than the non-injected leg. But interestingly, the other leg is also getting bigger. This is pretty
Mm.
cool. You're
Mm.
growing muscle in a 70-year-old human without exercise.
Yeah. Yes.
It's
So
pretty wild.
it is. Are you looking at the knee, the arthritis in the knee? Is that improving, or is that something you're looking at?
We are not treating knee osteoarthritis.
Right.
We are simply using it as an immobilizing event.
Hmm.
We would use casts, but not a lot of 70-year-olds are out playing soccer and break their legs and arms. So
So
we're using older
right.
people with an immobilization injury as the knee osteoarthritis. Interestingly, um, of course, we're using very sensitive MRI to measure the volume
Mm.
of their muscle. It's not just outlining with crayon on a picture. It's a very exact ways of measuring the muscle fat and the muscle volume and circulation and everything. But we also captured the patella, the kneecap. That's an
Mhm.
isolated bone. It's just sitting there connected with cartilage. Under an MRI, it looks like a floating bone. And, very interestingly, we're seeing some increase in bone density in these people as well. So I want to caution that it's early days in that we've
Mhm.
only got a handful of patients. There's only 9 humans in our entirety of our phase 1/2A clinical trial. We're primarily testing safety. That's the phase 1 portion.
Mhm.
But, this being the 17th clinical trial that I've run with my teams, we're quite good at it. And we talked to the FDA and asked, while we are subjecting humans to this as a safety, please let us, in a very articulate manner, determine whether it works. And the FDA is a bunch of very smart people that work very hard. And they agreed and allowed us to capture these multiple measurements of efficacy. So it's been very fun to see the, pain go down and the inflammatory markers go
Mm.
down, their bones getting more depth, their muscles are getting larger. We're not measuring metabolism, but I can say that the recipients are feeling very energetic. And so we'll have to see in the course of a phase 2 clinical trial. In a phase 1 clinical trial, you're not allowed to do double-blinded, placebo-controlled, like you are in a phase 2. For first in human, you have to give it to every single person and see if it's safe. And that's what we're doing as our primary goal. But we need to do the double-blinded randomized work, of course, to see truly what effect this is having. It certainly looks in these elderly people that have no business getting bigger muscles. Um, it looks like it's working, but to be completely sure, we need larger numbers of people in a phase 2 and a phase 3 clinical trial, of course. But that's the path of drug development.
But so the improvement, which is great, and across all these different aspects, uh, how do we know that it's related to the immune system? I mean, could it just be that the younger factors are just making the body feel younger and behave younger, or systemically
Sure.
rather than the
Yeah,
immune
that's a very
system?
good question. We take the blood from these humans that are in our clinical trial receiving Immuna every 2 weeks for 3 months, and we take it before they are getting our drug. And for every blood draw, we assess the blood for 1,000 different things. And
Mm.
it is an immune profile panel. So we know for certain after they are injected with Immuna that their immune system is fundamentally changed. And we can see, dozens of factors going up and down and, the different families of extreme pro-inflammation starting to drop down in concert. So we certainly know that it has an immune modulating effect. We've also looked at epigenetic profiling, and we can
Mm.
see that we're turning on and turning off different families of gene expression. So, for example, in the muscle or blood, we're seeing an increase in pro-regenerative and anti-inflammatory gene expression, and in our treated humans, and then a suppression of anti-regenerative and anti-inflammatory markers. So we're kind of, it seems to be pushing down the expression of genes that are associated with degradation and degeneration, and pushing up the genetics that are related to regeneration and anti-inflammation.
Have
So
you
it
ever
looks like it's
looked?
doing the right thing. And
Yeah, I mean
yeah, it looks like
it
it's tweaking
does.
the right system.
Yes. Have you ever looked at epigenetic age, either in kind of the mice or the people?
Yeah, certainly you know, the development of biological age clocks has skyrocketed. Just a
Hmm.
few years ago, I remember seeing 7. I think we have over 100 now. So a lot of markers that have done, probing of multiple immune system markers with biological age. And what we're doing is taking a much more detailed and scientific approach to that. It's not our intent to develop a little widget that you can drop your blood on and see what your biological age is. That's other people are doing that, and they're doing a lovely job of it. We use quite a bit more markers, 1,000 markers, not
Hmm.
5 or 6 that they target as a surrogate. Um, and we certainly see what is absolutely coincident with the reversal of biological age, but in a much more detailed manner where we can tell you precisely, is the IL-6 pathway, the TNF pathway, the TGF, the IL-10 pathway, is it going up or is it going down, and how long, and what are the circumstances of the human precisely? Are they getting better? And is there, the rest of their immune system working in a concerted manner? And this much more broad, deeper view that we're able to do with 1,000 marker tests, much more expensive and not, viable really for everybody to be looking at biological age with. These much more thorough tests show very similar things in that the genetic expression does seem to be reversing biological age.
So if you took a slightly longer look, so phase 2A now, phase 2B, phase
We're
3.
doing the phase 1/2A. That's
Yeah,
just finishing up, and
yeah,
we'll be beginning soon, probably August,
yeah,
September, our phase 2.
right. And then, so when do you think you'd get into phase 3? I mean, is that something that you can speculate on?
If there's one thing I've learned, trying to predict what the FDA wants to do in their timeline is very tough, and it's also
Okay,
foolhardy.
okay.
We've done a lot, and we do work with regulatory experts and very closely with the FDA. If if um uh if everything goes as we expect it to go from the norm of FDA behavior and application processes and policies and deadlines, then we should be in the third or fourth quarter of this year beginning our metabolism clinical trial. In 1 year thereafter, we'll hit an interim data, which will be a tremendous amount of irrefutable, double-blinded, randomized human data. That's about, a year and a few months from now. 1 year after that, the clinical trial will finish in its entirety, and we'll have a complete phase 2 data set. I'm exploring other things now. We may actually run multiple phase 2 clinical trials, some of which are faster than others, in multiple manifestations of immune-mediated aging. And we have a lot to choose from. We have beautiful, beautiful preclinical data on muscle and metabolism. Both of those are published, but we also have excellent data on neuroinflammation, on arterial stiffness and wound healing and the adaptive immune response. So there are several other areas that this could go into in a, it's expensive to run these human clinical trials, but I'm pleased to say that the interest is phenomenal. The
Mhm.
marketplace right now and the economy is very good. Let's hope it stays that way. But for the time being, it does seem to be a very fundable story that's getting a lot of interest. So I may be able to raise the capital in order to try this in multiple human degradations rather than just simply muscle atrophy and metabolism.
right? Do you see kind of the endpoint as it being systemically, so rather than fixing muscle atrophy and sarcopenia, it's like systemically being able to, I don't know, I don't want to use rejuvenate, but to make more healthy
Yeah.
the whole human.
That's my intent. You know, my team and I at Immunis are intent on a prophylactic approach. Really, we're meaning this to be used in a prophylactic manner so that you keep your immune system strong, so that you don't get these immune-mediated declines. I started off our discussion by saying I was initially drawn into this by trying to help cancer patients not get cancer,
Mm,
because there's no cancer patient that had cancer that wasn't immune suppressed. Like immune dysregulation is necessary for cancer to occur. I started out by really with the team looking at how do we keep the immune system healthy? And then it became very apparent that if you keep the immune system healthy, you stave off a whole lot of death and demise in a lot of different ways. You're going to not die of some bugs that otherwise would have killed you. You're going to not get cancer. You're not going to generate allergies and just tie up your immune system. There's a bunch of things: arterial stiffness, neuroinflammation, adaptive immune response, muscle, metabolism. All of those things are very fundamental to your quality of life. And they also have downstream effects. Think about your ability to be motile and functional
mm,
and competent from a mechanistic, a motor point of view. If you're 85 and you can walk up and down stairs and walk around the block and cook yourself food and take care of your own body, that your quality of life is extraordinarily bettered. So it's really my goal to develop a prophylactic where we can see a physiological function that's causing in this very dirty society that we live in, and taxing immune systems right, left, and center. If we can better our immune system in order to keep ourselves a little more resilient, keep ourselves stronger, keep our metabolism higher, and that combined with a lot of behavioral adjustment on the part of a populace that is
mm.
becoming much more educated now, I think we've got a good shot of really improving quality of life in a very large way to a very, very large percentage of the population.
Right. So when you get to a certain age, I don't know, 60 or 70, then you would start like taking this, as you say, prophylactically, like ahead of
Yeah,
time.
I would think so. It's certainly, in human clinical trials, you're not allowed to work on healthy normals. You have to
Right.
work
Yeah.
on worst off of the worst of any particular thing. You need an indication. So, we're tackling people with trouble, right?
Yeah.
They can't they can't locomote normally because they're older and they have knee osteoarthritis. This is fantastic for autoimmunity, right? This is
Yeah.
fantastic for in any type of immune suppressed environment, autoimmune environment, immune dysregulation. But for a healthy, normal, goodness, keep my immune system strong.
Mm.
There's a lot of ways you can do it right now, but most of us don't do that. So,
Yeah.
you know, we need to work in keeping our immune system strong. And as you get older, there's no amount of work that'll keep it strong. You need some help. And I think Immuna could be the help.
Yes. So kind of taking a step back. So what do you think? I mean, think about aging in general. So we think about, like
Mhm.
why do we age? So what do you think? What do you think using the secretome and the effect it has so systemically would kind of say about that? Because it's just well, it's a bunch of factors, but it's just a small piece, and yet it seems to have such a systemic impact.
Well, I'm not sure I understand your question
Okay.
entirely, but
Mm,
what comes to mind is that humans are a very long-lived
mm,
species. We're one of the longest-lived species. And as a result, what happens from a biological point of view is that there's an unintended consequence, which is cancer. So we have
mm,
to, are living so long, we've got to replace 100% of our body, except our brain and spinal cord, about 14 or 15 times during our, 60 or 70 years. That's extraordinary. And because we're so long lived and we have to keep dividing and dividing and dividing sometimes messes up. And you get cancer. And now, through evolution of vertebrates with longer and longer lives, there's been a more complex immune system developed in order to police all of that. The longer lives mean that also that we experience more bugs. So we have to maintain an immune response. And right now, somewhere around, 40 to 80 years old, I know that's a big swing, but it happens in all of us in that time period. Our immune systems just kind of cack out. Our naive immune system there's no longer any naive immune system anymore. We've encountered so many bugs that those naive cells that are waiting around wondering what to do, they get told what to do with SARS-CoV-2. And now you've got a whole bunch of immune memory cells in you for that bug. You've got a
Yeah.
whole bunch for every flu and every sickness that you've ever had. And our immune systems just run out. And, and unfortunately, that's the trigger. That's the domino to most of the manifestations of aging. And I know not everyone feels that way. I've come to learn that, and it's a driving reality that I see that is under-recognized in the community, generally speaking. Immunologists get it, but not a lot of other groups. And it's just a growing awareness that many of the manifestations of aging are immune mediated, and your immune system is very, very difficult to modulate because of its redundant nature. So it takes a knowledge of that. And then for me, about 15 years to work with my team to then develop, um, a drug to modulate it, and also to work with regulatory agencies to get regulatory agencies to accept a multi-active drug, which they never have before. So there's a lot of firsts
Interesting.
in this story. It's been a political one, it's
Hmm.
been a regular one, it's been a big money one, it's been a donor one, it's been a hardcore science, and now medicine. So the fun things in life are not easy.
Yeah. Yes. So Dr. Diamanda, well, it came from Ray Kurzweil, right? The longevity escape
Yeah,
velocity. I just wonder
yeah.
what your thoughts are on that. I mean, how far are we away, or do you think we're there already?
You know, the concept of longevity escape velocity is such that, for every year that goes by, we're getting closer to living that, extending our lives by that extra year. And at one point we will extend life so much so that during that period of time, there's going to be another life extension. So I think we're close, to be honest with you. We're nowhere near close for the general populace, but we're very close in C. elegans in worms. We're very close in little tiny critters. Now we're starting to see these manifestations with our clinical trial in humans. So it's coming a long ways, and I think it's going to take a very, very much better understanding of the immune system. Hence, my willingness to lead the Human Immunome Project to do a global
Hmm.
fingerprinting or sequencing, if you will, of humanity's immune system. It's no less, and indeed, it's a much larger endeavor than the Human Genome Project. And but we need that information in order to properly know how to modulate. This is one of the early um, modulators of the immune system. There's going to be
Hmm.
more slightly different secretome approaches, a very, very scientific dissection of the 440 factors, withdrawing some, doubling up on others to find out which are the most important, and then perhaps using them to discover very refined targets. When you think about it, we kind of have a shotgun approach here with a
Hmm.
very natural, um, you know, replenishment of what you had when you were young. So we're not messing with it. We're just putting the whole thing back in there, your entire secretome that you had lots more of when you were younger. And whichever one is responsible, probably dozens of them that are responsible for maybe the metabolic benefit or the muscle benefit or the arterial. We are first going to get this drug to market. I'm really with a team at Immunis. We're focused on getting this drug into humans in the broadest way. It has tremendous benefit and no side effects. Let's get it out there and help a bunch of people. And as a first order of business, and then along the way and thereafter, refine, 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 and maybe develop along the way, a lot more targeted finesse rather than a broad approach like this. I have a sneaking suspicion that nature does it a lot better than we ever will. So this broad, all natural approach that we're taking now, simply letting young precursors put out the full secretome, modulating the human immune system like you do, I think that's probably going to win the race. So I think we're doing the right thing right now. But we're a bunch of geeky scientists, so we're going to dissect the crap out of this thing and figure out every little on and off switch.
Right. Yeah. I mean, there are a number of interesting technologies at the moment, partial reprogramming and so on. But what you're working on seems to be like the closest to actually being releasable, and it's great
Yeah,
that
thank
you,
you. It's in humans. It's not a
yeah,
lot that is.
and it's in humans, and you've got it the farthest so far, and you already have a history of taking drugs through these trials, which is really good to see. So that's all really encouraging. So one last question.
Yeah, it's
What
exciting, very exciting.
it is.
Sure.
So what are you willing to share your personal protocol? I mean, what is it you think about? If you want to kind of take advantage of the longevity escape velocity, then you need to maintain your health till we get there. So
Yeah.
can you share your personal protocol? What is it you think about?
Yeah, sure. I'm happy to. It's easy. It's you'll hear me say this and you'll think, "Oh my goodness, I could do that." But I challenge you to actually do it. It's incredible how as humans we romanticize and don't get around to the practical stuff. I have a very simple regime, but I am rigid in it. I am
Mhm.
rigid. I follow the rules I set for myself. And it's basically sleep, laughter, exercise, and healthy eating. And I do a lot of those things. I really spend a lot of time making sure I can sleep. And that's glib. I've tried a ton of things. I have a lot of behavioral practices like no electronics after 7 p.m. And my goodness, I am the busiest person I know. It's really hard to do. But
Mm.
I make sure that those four areas, which are four irrefutably documented, scientifically and medically proven ways to improve your health and healthspan and lifespan, is sleep, laughter, incredible immune stimulator laughter, exercise that's moderate and most days, and very, very healthy eating. Add to that a ton of diagnostics, but you have to remember I'm in the healthcare business and I'm a geek. And so I dissect myself every 6 months. Basically
Mm.
spit, blood, stool are excellent ways to start that is very, very approachable. It's very affordable. Everybody can, almost everybody can do it. It's still in the thousands of dollars per every 6 months, maybe three, sometimes as much as seven. But getting spit blood stool done, and you know a lot about yourself. What you will get from that helps you adjust. Because I'm in my 50s now. Of course, I'm looking at all the things that hit 50-year-olds, and with spit blood stool, you can tell whether you're one of those 7 out of 10 humans walking around with H. pylori burning a
Mm.
hole in your gut, changing your gut biome and giving you allergies. You can find out if you're one of those humans that has coronary heart disease, like a lot of us have. You catch that stuff early, you don't die of it. You catch it, you treat it, and you don't ever know that it was there, besides a bunch of little numbers on a page that came from your spit, blood, or stool. So I do that. I behaviorally adjust, and I have a supplement regime that helps me get rid of stuff and change and slowly tweak. You don't have to be in the healthcare business like me. Just get it done, and by a nutritionist group or go on the web and find it, and then you can up it if you want into greater levels of diagnostics like MR whole body MRIs and things like that. I do it all just because I'm a healthcare geek and love that stuff. But it's really the very basics that besides the diagnostics: sleep, laughter, exercise, and healthy food. You do those things, you're going to be staving off a whole lot of evils.
Interesting. Yes, excellent. I mean, would you be able to share one or two, well, your top supplements? What do you take?
Yeah, yeah, absolutely. So, like most Americans, I had high H. pylori, and so I
Mm,
take HCl, just basically acid pills every
mm.
meal. And now I've almost eliminated H. pylori. You know, H. pylori is associated with cancer and also of the guts, and as well as ulcers, and it completely changes your microbiome. Um, I've cut down on alcohol quite a bit, and as a result, my sleep is phenomenal now. And that has all tons of downstream effects. But I'm being very careful of probiotics and acid to get rid of my um, H. pylori almost gone, so almost going to stop that, and a few other immunomodulators that just tweak your immune system a little bit, keep yourself a little bit more balanced. So I do a handful of supplements every meal, and I change them out every 6 months in order to just slightly tune it to what my body's showing at the time.
Excellent. So, is there anything else you would like to talk about? Anything that we you don't think we covered?
Well, I'd only throw one caution out
Mm,
there for your audience. I've been a stem cell researcher all my life and all my professional life, and I can tell you that when I started, nine out of excuse me, 9 out of 10 stories were snake oil, and now 9 out of 10 stories are still snake oil. The stem cell sector has had it's an incredible marketing angle, and it's been abused. So you have to be very careful in Healthspan and lifespan and longevity. I'd say 95 out of 100 things I'm hearing are absolutely nonsense. Although there's some very, very good work out there. Ours, a lot of you know, epigenetic reprogramming work that's working very good, not quite translatable to humans, but looking very, very attractive. There's some really good stuff coming out, but I'd say 9.5 stories out of 10 are absolutely ridiculous. So you do have to be very, very careful. And is it being run by the FDA? There's a big one. Why not? If you've got a drug and you can run it through the FDA, there is absolutely irrefutable evidence that it reaches more people faster than if you don't. The only reason that you don't run through the FDA is if your drug can't do it. Like, it's either not safe or you can't make it without batch to batch variability. So you don't know if you're going to overdose somebody and give someone too little. So, really be very, very careful about, what's real and what's not. And a good measure is the United States FDA. Another measure is just type in the researcher's name and say FDA cease and desist, because the FDA puts out about 300 cease and desist letters every year. And I don't know how much they do every year, but they have over the last couple of years, around 300 a year. And if you type my name in, there's no cease and desist letter from the United States FDA beside it. You should just be careful about, you know, who has one, why do they have it? Is the FDA regulating this work? You're going to see a lot more nonsense because the market is just so massive. It's driving a lot of good players and it's driving a lot of bad players to the sector because, the demographics of the world we now have more older people than we've ever had on the earth before, and it's going to get 10 times worse over the next few years. So we
mhm.
do have an aging population. That's where the money resides. And a lot of bad players are going to be trying to grab that. So it's an interesting field. Look out, use diligence.
Okay, and on that note, so thank you so much for joining us.
My
It
pleasure, Rick.
was it has been great talking to you.
Yeah, really fun to talk to you too. Thanks for the exposure and all that you're doing. And everybody out there, stay healthy, eat good food, and do a bunch of laughing.
Okay,
Thanks very much.
thank you. Yeah.
All right, bye-bye.
Okay, bye.